Good morning, everybody. Welcome to our this week's edition of Micro Seminar. We hope everyone is faring well. We hope you're healthy and safe. It's my pleasure to introduce to you today Dr. Will Shoemaker, who's just finished his PhD at Indiana University with advisor Jay Lennon. He's been working on microbial dormancy there, and among other things, transitioned from a primarily field-oriented, field ecology focus to experimental evolution. Prior to that, he did his undergrad at James Madison University, where he was studying amphibian microbiomes in Reed Harris's lab. And he's moving on to come join us down here in wonderful sunny Southern California with a postdoc in Nandita Garud's lab at UCLA. And so without any further ado, I'm going to let him tell you about his work. Thank you for being here, Will.
All right. Thanks, Cameron. I'll have to get used to the whole doctor title. Okay, so let's see. Share here. And okay. Cool.
Looking good.
Put this in the corner. Okay, so today we're going to talk about some experiments that Jay and I have been running in his lab to look at the basically the population dynamics and ecology and evolution of microbes in a very energy limited environment. And so, like, I usually like to start talks by talking about the just sheer diversity of microorganisms on the planet. By a number of metrics, they're conceivably, sort of the winners. They have the highest global abundance. If you're, sort of lenient on definition, they have the highest number of species. They've colonized virtually every conceivable environment on the planet. And that includes environments where the concentration of resources is very low, to the point where growth, in addition to maintaining cells, is very difficult, if not impossible. So this, can occur for a wide range of environments. One of the more probably popular ones are the deep sea sediment, where a lot of work has been done to characterize the sort of deep biosphere in there, where you've just got crushing pressures, low temperatures, as well as a very low flux of nutrients going through in the aging sediment. Two, very different environments, but similar in the lack of energy, which would be, deserts, where microbes have to survive the sort of boom bust cycles of periodic rain that can occur with that spaced by large periods of drought. To environments that, at surface or may not look that energy limited, or at least not as popular as deep sea or the desert, but mountain runoff from glaciers is one example where you actually have really low concentrations of resources where that can limit the growth and reproduction of microbes. And this isn't just in environments which are relatively difficult for humans to inhabit. Energy limitation can in fact occur in the built environment as well. And one really good example of that are hospitals, where microbes, disease-causing microbes, basically have to be able to survive on a sterile or at least desert-like surface for extended periods of time until they're able to get picked up by another host where they can continue to grow and reproduce. And it's not just that the environments are diverse. Microorganisms have evolved a diverse set of strategies to be able to survive these extreme energy limited environments. So top row here, top left, classic example, bacillus and many firmicutes can form protective endospores that allow them to survive extreme, environmental factors, in addition to just low concentration energy, such as gamma radiation, vacuum of space, intense cold. Top left is just single cell electron microscopy image. Top right is a stained image. Two microorganisms that don't necessarily have to form a new structure, such as mycobacterium in the bottom left, that can just enter a metabolically dormant state where the tool to survive in a human host for extended periods of time until it can, be resuscitated. as well as other interesting structures such as the formation of filaments by many cyanobacteria to survive desiccation. So there's this wide variety of environments, this wide variety of structures, but it basically, the population dynamics are very similar. Microorganisms, at least in their growth, can be generally characterized by this growth curve where they start out, you have this lag period where the rate of growth is very, very low, and then you enter this exponential rate of growth until some substrate runs out, and then you enter this stationary phase for an extended period of time, followed by extended death. And this increasingly characterized phase of the curve known as, or characterizes the long-term stationary phase, where it appears that death rate, or at least the net rate of growth, appears to decrease over time, and microbes can survive for extended periods of time in this phase. And so the first three are, very standard. The second two here been, increasingly characterized over the last 20 years, but there's still a lot of basic questions to answer about what's going on here and how that allows for, and how microbes survive under these demographic scenarios. And so this is sort of the regime that Jay and I have been working in for the past, well, since he's been doing this before I got here, but that I've been doing since I got at Indiana University about 6 years ago. And so these are regimes where the net rate of growth is extremely low. The experiments take a long time, and so you really need you need to run out your experiments for a long time scale in order to sort of characterize these dynamics. So today I'm going to talk about two basic questions that I think characterize a lot of what I've done here at IU. The first is a very sort of general, what happens to the population dynamics of microorganisms as the energy runs out. And this is going to be a bit more of an organic question, because we're going to start out with sort of a fairly straightforward experiment that eventually some new interesting dynamics came to play, and it became a bit more complicated in a good way. And then the second is a bit more straightforward. We're just running evolution experiments to determine how microorganisms evolve under in energy limited environments. So when you've got these energy limited environments where things aren't really rapidly dividing, birth events can't occur, your null expectation is that the population is decaying at a constant per capita rate per time, right? So you've got some change in the number of cells that are viable, and that's just equal to some net rate of growth in your population times the number of viable cells at that given point in time. So the net rate of growth, just birth minus deaths. And so since you've got virtually no resources in your environment, you can say that there are way more births than there are deaths in your population, right? So D is much bigger than B, and you can sort of say that, okay, the change in the number of viable cells over time, that should just basically be reflective of the death parameter, the death rate. And then from there, you can get some interesting quantities of interest for understanding how microorganisms survive and potentially relate them towards natural environments, such as the mean time it takes for a cell to die, right? So this is our basic framework going in, the quantities that we want to try and estimate. And we were trying to do this using this long-term, what we call dormancy experiment, but energy limitation experiment, where you've got 21 taxa in your experiment, and with a total of 96 separate replicate populations, right? And so what you have is each replicate population is run out for 1,000 days as these survival curves, where we first just harvest the active cells from a standard culture. We pellet them and wash them 3 times, so you removed any residual carbon that might be in the media. And then you resuspend those washed cells in phosphate buffered sealing with no resources at all, and just count the colonies over time, right? And so our null expectation here is that we'll just get some first order decay of population size with respect to time, and then we can calculate death rate and get the distribution of death rate across taxa, and get some interesting, get some quantities of interest. So what we find is that really only one taxon actually met that expectation that we had going in, and that's the micrococcus strain that we're working with, where in a semi-log scale you would get that nice linear decay with respect to time. So you can't actually apply this straightforward approach where it's just a simple linear model, and then you get some nice estimate of the death rate in the population. However, all the other taxa had this curved pattern where the curve extends out in a way which indicates that the net rate of growth is increasing with respect to time. And so the vast majority of taxa had this sort of like a little bit curved, it's an obvious curve on a visual scale, but it didn't seem like extremely drastic where it compared to, say, Bacillus, which was a clear outlier where just the number of cells initially plummeted and then had this basic steady state occurring, which is no doubt due to the fact that they're able to form protective endospores, at least in part. Right. So the null model doesn't work, and the question was, how do we analyze these curves, right? Because we didn't have a mechanism in mind which we could build up a model and fit to the data. So we know that the change in the number of viable cells is greater than the initial change after some number of days, but we don't know the actual form of that model. And so instead we're trying to stay agnostic by performing survival analysis. And so we chose ultimately we did a number of approaches, but we eventually landed on the Weibull distribution for its flexibility. And so basically how this works out is that there's some initial rate of death in the population, right, when you start, right? And so if the initial rate of death does not change over time, then you just get, the integral of that differential equation in the previous slide, which just means that you have this exponential decay on a linear scale. And so all you're basically adding with the Weibull distribution is a parameter to control the shape of the number of cells at a given point in time. And that's this parameter k here. And that reduces to an exponential model, right, if k equals 1. So it's a fairly straightforward approach used in a ton of medical type survival analyses. And so we chose to just use this and apply it to our experiments. And so it works pretty well. I mean, so with Micrococcus, we know that just from looking at it, okay, there isn't really much curvature, and so we fit the model, and we do see that there's a very slight deviation where the k shape parameter is slightly less than 1, but it's pretty much well described by an exponential, and if you do like a likelihood ratio test, it doesn't come out as different. Versus all the other taxa we examined where Weibull model does a good job describing the survival curves. Bacillus, it's getting close to breaking the model because it's just such a sheer drop, but it's fairly decent, at least been able to get get a fairly decent fit out of Bacillus, right? And so what we can say is that the more that k decreases, the more that the shape parameter decreases, that corresponds to a greater increase in the net rate of growth in the population over time. Right. And so the question from here is, why is this net rate of growth increasing over time? What's the actual driving mechanism? And we had some hypotheses going in, but a number of lines of evidence led us to the conclusion that this increase in growth rate with time is primarily driven by cellular recycling. Right? And so the way this works out is that you've got some initial number of viable cells in the population, large number of them die, and then those dead cells decay and lyse and release some substrate into the environment. And then as that substrate accumulates, it can be used for growth. So as cells die and release the substrate, then eventually there is this increase in the net rate of growth where that occurs because cells are taking in that substrate. So that's the actual, believe the mechanism that is causing this increase in the net rate of growth over time. So key point here, when you start out, there is no substrate in our experiment, right? Because we just take cells and we put them in buffer. So the initial death phase is just this linear decay. And so any future change in the net rate of growth depends on the initial number of dead cells. That's the prediction going in. And so there's been a number of, a few models that have incorporated necromass consumption into these sort of dynamics through as a system of ordinary differential equations, right? And so you examine those models and you can see that okay, there's very few initial number of deaths here in the red, more in the yellow, and the most in the blue. And as you increase the initial number of dead cells, you eventually have this greater increase in the net rate of growth over time. So this survival curve becomes increasingly curved as you increase the initial number of dead cells, which is just your initial estimate of the death rate times the initial number of viable cells in your population. And so if we calculate, so you have this prediction here where the initial number of dead cells should be negatively correlated with the shape parameter, right? Because since you have fewer and fewer dead cells initially, there's less and less change in the growth rate that can occur, versus at high concentrations, high numbers of dead cells, you have a greater change in the growth rate. And so this holds. We are able to get this by just our empirical estimates from plating of the initial number of viable cells and the initial death rate that we obtain by fitting the Weibull model. And if we just simply examine that relationship with the shape parameter, we see a fairly straightforward linear relationship. Now, the slope parameter doesn't have has a meaning in terms of units, because this shape parameter here is just sort of this parameterless thing added to an exponential to allow for curvature. But it does correspond, this relationship does correspond to us being able to say that the change in growth rate is proportional to the initial flux of dead cells, a primary prediction of necromass consumption. And so some additional evidence for recycling. We find that dead cells don't accumulate over 1,000 days from just microscopy work. From mass spectrometry, we find that the concentration of amino acids in the cell-free supernatant is below the detection limit, right? And so the dead cells have to be going somewhere. And so since we don't count dead cells in the environment and we can't really detect their components in the media, this implies that cells are being recycled. And there are a few more subtle lines of evidence, but some interesting ones that correspond to some interesting physical models of growth. So one thing we find is that cells that have a greater increase in growth rate, right, the shape parameter is consistently less than 1, spend less time in lag phase. And so this relationship falls out as a basic prediction of kinetic models of microbial growth, where they're able to basically examine how lag time falls out of simple kinetic models. And this relationship they observe is consistent with the fact that necromass recycling would require some amount of kinetic activity to occur within the cell to allow for the net rate of growth to increase. So to examine how the net rate of growth is increasing, right, demographically speaking, you can either have an increase in the net rate of growth by having fewer deaths and more births. And that's actually quantifying births, especially in like a long-term experiment such as this at this scale in a time result way, is fairly difficult. So we have a few indirect approaches that can indicate the demographics in terms of birth and death. One thing we find is that there's no relationship between the shape parameter and a coverage-based proxy of birth rate, the index of replication, which is just basically the slope of the distribution of coverage when you sequence the population and map it to a reference genome. And if we look at the mutations in populations that we get by just doing a bulk sequencing from the end of the experiment, we find that there are hundreds to thousands of mutations that occur within each population, but they're all at a decent frequency, but they're all hovering. The maximum frequencies across populations hovers around approximately 0.3 to 0.6. So there are no substitutions occurring in any population over 1,000 days, right? And so the lack of substitutions tells us something about the number of generations that occurred over 1,000 days, which in turn tells us something about the birth-death dynamics of the system. And if we do examine the site frequency spectra of all these populations and examine, what proportion of mutations are or polymorphic mutations are non-synonymous or synonymous. We do find evidence of negative selection across taxa with, very little evidence that there is any adaptation occurring, which that is what you would get if there really weren't many opportunities to increase fitness by acquiring mutations that lead to a higher birth rate. So this is one line of evidence that indicates that the growth rate is primarily driven by decrease in death rate, but we can tease a bit more information out of these mutation frequency spectra, right? So basically by doing some back to the envelope calculations here, we have a table with all the taxa that had enough biomass that you could actually sequence them all, and the number of mutations in it, their mean allele frequency of those mutations, and the maximum frequency. And so if we assume that birth events occurred in the populations as a just binary branching process, we can calculate we can do some back of the envelope calculation of the minimum number of birth events that occurred in the system, then as well as some other quantities like the time for a birth event and things like that. And what we basically get for the minimum number of birth events, we get about 10 to the 4 to 10 to the 6, which does seem high, but if you put that in contrast with the change in total population size over 1,000 days, or try to see how like the extent that these birth events can explain the total change in population size, it's a very, very small fraction. And so basically you can end up with birth events contributing approximately 10% to the demographic change that occurs over 1,000 days. And so this is basically just us pushing what you can do with any sort of frequency data. But just doing some back of the envelope calculations, we are able to tease out a little bit of information on what exactly is going on. And it's consistent with this interpretation that the increase in the net rate of growth is primarily driven by a decrease in death rate. So that's actually nice, right? The fact that death is the driving demographic factor in our system means that we don't necessarily have to worry about the likely complex birth-death equation that would ultimately govern the system. And we can just use our survival curve and say that, okay, deaths are much, much more common than births. So to get some quantities of interest, we can simply ignore the contribution of births in this experiment. And from there, basically calculate the mean time to death of a cell over 1,000 days. And so this is just basically taking the mean of the Weibull distribution fit parameters. And from that, we find that the mean time to death of a cell over 1,000 days is approximately 17 days across all taxa. So this number here is the mean of means, right? And that's shown by this gray line here. And if we examine the distribution across taxa, there is variation across taxa in terms of the time to death, with a range of 1 to 1,000 days, of bacillus being a clear outlier. Now, it's difficult to detect extinctions, but if we sort of operationally defined extinction as a concentration of cells so low that we can't detect it through plating, right? We can establish a minimum frequency for cells in the population relative to when they started. And from that, establish a time until a population is effectively extinct, at least from the observer's perspective, right? And so that time to extinction ranges for about 1 to 100 years across taxa, with bacillus of course, being a clear outlier where its time to extinction is about 100,000 years, which is sort of pushing it, since it does seem to be flat and there isn't much sign of decay in the actual empirical data. But at least for the remaining taxa, we're able to establish a time to extinction for this relatively simple environment. So as a brief recap, we determined that the growth rate increases over time under energy limitation, and that's driven primarily by necromass recycling. This increase in growth rate is driven primarily by a decrease in death rate, right? And the average, we can calculate the average time to death and get the variation across taxa, and at least in the taxa we examine, we see that approximately varies over 3 orders of magnitude. With Bacillus is a clear outlier, and while there's vegetative and spore-forming cells in those populations, it's, the fact that Bacillus is able to survive so long is clearly in part to the fact that it can form endospores, right? It can form this reservoir of dormant individuals that are really protective. So a seed bank. So from here, we were sort of asking how do microorganisms evolve under energy limitation? So we sort of just did this experiment where it could basically be viewed in a way as one really, really long transfer, 1,000-day long transfer. And so just taking a sort of experimental design and shortening the time, but doing multiple transfers effectively and seeing how microbes evolve over time. Right. And so one thing we really wanted to do was incorporate dormancy. So, large numbers of microorganisms survive in nature by entering a metabolically inactive state where they can form a reservoir of genetic diversity. There's been some evolution experiments, but just simply comparing dormancy capable and incapable populations in evolution experiments has not been done before. So dormancy basically, you've got standard birth death events happening in some part of the population, but individuals can also enter a dormant state and resuscitate, which is analogous to migration between two populations, with the exception that this death rate is likely much, much smaller than the death rate over here, and that you can't have any birth events occurring for dormant individuals. And so basically some simple predictions that we wanted to test relate to the to the amount of genetic diversity in dormancy capable versus incapable populations, right? Where dormancy capable populations tend to have higher levels of diversity. The dormancy incapable, they tend to have this longer time to fixation for the frequency of alleles, right? And that's simply because you've got this sort of other population in your population, right? Where individuals can enter that dormant state, they don't increase in their mutations, don't increase in frequency until they resuscitate and are able to, divide again. And so for populations that can form seed banks, we want to ask, do they have higher levels of genetic diversity over time? Do they decrease the rate of evolution? And third, slightly more subtle, but do they change the direction of evolution? And that's in the sense of, do certain mutations do dormancy capable populations acquire mutations in different genes than dormancy incapable populations? So how we did this is that we engineered a form of Bacillus subtilis that can't form protective endospores, and we did that by removing the master regulatory gene, SPO0A. And so just to give an idea of what sort of happens when you do that, we've got these experiments where you basically starve the population for 1, 10, or 100 days. And if we look at the fitness of the SPO0A relative to the wild type, we're able to see that at 1 day, they're about the same. There's really no fitness cost for removing this master regulatory gene. And at 10 days, interestingly enough, the knockout has a fitness advantage over the wild type, right? So something's going on where the wild type is able to have this higher, or the knockout's able to have this higher net rate of growth after being in this cold culture for 10 days. And as you get to 100 days, at that point, anything that you can use is to grow and maintain the cell is gone. And so you have this basically winner. The winner here is the wild type cell. So our experiment here is basically, you've got transfers going on every 1, 10, or 100 days for your replicate populations. And sort of just coarsely, if we were to map it on this original conceptual demographic model of what's going on, we could say that, okay, you enter stationary phase probably at the end of this 1 day period. You're in this stationary or death phase at 10 days, and then you're really, really, really into this long-term stationary phase after 100 days. So this is sort of a coarse mapping. We don't have exactly measurements that allow us to point to where it is exactly on this map, but just to get an idea of where these treatments stand with respect to sort of standard sort of growth curve dynamics. So what we had were 2 strains 3 treatments, 1, 10, and 100 days, and 5 replicate populations, giving a total of 30 populations. And so this was part of a larger energy limited experiment that Jay and I worked on. And so this experiment was primarily conducted with me, a former technician, Evgenia, and a former technician, Kenzie Gibbons, who's now in Matt Hahn's lab doing complexity work. So basically the experiment went on for 1,000 days for the wild type, and we ran the knockout for 700 days. And so this is effectively a 3-year-long evolution experiment with manual transfers. So 30 populations, every 100 days, we collected biomass, we did glycerol stocks. So we ended up with a total of approximately 300 DNA extractions. We did a mixture of in-house Illumina library prep and then eventually working through Indiana University's Center for Genomics and Bioinformatics, aiming for approximately 100-fold coverage. And so just to clarify, what we did was we did bulk DNA extractions from the entire population and then built a library out of that bulk DNA extraction. So this should contain DNA from the entire population. From there, we would try to estimate allele frequencies. And so this is, some people call it pooled population sequencing, others have called it in this setting, metagenomic sequencing. But basically the goal is to get the frequency of mutations from the whole population without having to sequence individuals. And so we cleaned the data by adapting previously developed protocol by Ben Good and Michael McDonald for their 2017 Lenski experiment paper, and using their basic analysis approach to distinguish what we would consider real mutations from mutations that are likely to be errors, sequencing errors, using a permutation-based approach. So that way we can get some estimate of the mutation frequency at each time point, right? So, and so just to give an example of the sort of data for like the unfamiliar viewer, you sequence your whole population, you get these mutation frequencies back, right? And so you don't know what mutations co-occur with one another because you don't have that linked read information that you would get if, say, you sequenced every individual separately. So we don't have any information on linkage. But we know, basically all populations start with initially 0 mutations, and then mutations get acquired and they increase in frequency and can go to fixation. And you can't really tell whether or not something like, say, clonal interference is going on just from the raw data. You'd have to do some sort of subsequent analysis, but you can see some obvious patterns in the data, like this red mutation here, it's increasing in frequency. It's increasing in frequency at the same time as all these other mutations, right? And so one of these is likely to be the driving mutation that has that sort of large fitness effect, and the others are sort of along for the ride, right? Since recombination since this is just that recombination isn't kicking them off of that lineage. So those continue to go on and increase into fixation together. So if we look at just these broad allele frequency trajectories for different populations and different treatments, we are able to see that the wild type has some sort of like qualitative difference in terms of the allele frequency trajectories relative to the knockout. There's much higher number of mutations in the population at any given point in time, consistently across 110 and 100 day populations. There's an outlier here on the right here, though we have reason to suspect that this is the result of it becoming a hypermutator. Right, and we also see that as you go from 1 to 10 to 100 day treatments, there's fewer and fewer fixations going on because there's effectively less and less generation since you have more and more time between transfer events. Right, and so if we sort of just summarize this and collapse all that information down to a single parameter, this sort of mutation estimate where you just sum up the allele frequency, the frequencies of all mutations in a population at a given point in time and look at how that accumulates with respect to time, we're able to see that for this top row here, yeah, the wild type that can form spores does have a higher amount of genetic diversity in the population. That sort of gets obscured by 10-day transfer regime, which is likely due to the fact that SPO0A has higher fitness, and so potentially is capable of more of more growth, more birth events, acquiring new mutations, as well as driving existing ones to fixation. But then we see this pattern again by the 100-day transfer regime, where there's a clear gap between the number of mutations for the wild type and SPO0A. And so we don't see effect really on the rate of evolution if we just take the number of mutations at a given point in time divided by the number of mutations at the previous time step and look at a log scale. They all tend to follow this similar pattern. So the actual quantity of mutations does seem to differ with respect to whether or not you can form a seed bank, but the rate of evolution, at least in the scale that we are able to measure here, doesn't seem to depend on whether or not you can form a seed bank. So something we wanted to look at basically is dig more into this allele frequency data and try to determine whether or not we can detect the effect of dormancy beyond just the simple how many mutations do you have sort of question, right? And so something that we wanted to consider was the fact that, okay, if you can enter a dormant state, right, you can form a seed bank. That just basically is a special case of demography, right? You've got multiple populations effectively going in your flask. Right, and so the fact that you've got multiple populations means that a given mutation in an individual can be in two different states. It can be growing or cannot be growing. And so if if you're unable to form a seed bank, you've just got this simple birth-death type model. And so as your environment changes, your allele frequency can change as it increases during, when you're capable of rapid growth, to maybe slowing down or decreasing in different environment. But the fact that you've got two demographic states here means that allele frequencies can effectively become uncoupled as, say, a mutation is stuck here in this dormant individual, and then some other proportion of those mutations are in the active portion of the population. And so what we basically find is that mutation accumulation curves asymptote after approximately 500 days, our initial plot. And so if evolution sort of stops at stationary phase, right, where you're not able to continue to, grow and divide and mutation frequencies don't really change that much, the number of mutations in the wild type should decay linearly with evolutionary time, in this case, generations. And so if we look at this here, we back calculate the number of generations that have occurred in the experiment and plot it against the number of mutations when they saturate. We see that a linear relationship occurring between bacillus for the bacillus wild type. And this dashed line is just sort of what you would expect if the slope was equal to 1. However, that's not the case for the knockout, right? We see this deviation from this linear relationship to the point where we can't even reasonably fit a linear model. And if you look at the residuals of the plot, we see that, there is very little deviation for the wild type and a much stronger deviation for the SPO0A. So there's some nonlinearity involved, which is consistent with the prediction that the ability to enter a dormant state basically is an evolutionary buffer. So we're basically just looking there at mutation frequencies. And so we want to be able to look at the mutations that actually, went to fixation and the mutations that went extinct. And so these states are effectively unobservable. They need to be inferred from data but that you've got some ancestor and the mutation can go fixed or it can go extinct. And so that's sort of been leveraged in Ben Good and Michael McDonald's paper as a hidden Markov model to classify evolutionary outcomes, right? Where you start as an ancestor, you're polymorphic, and you can be polymorphic again and again and again, and then until you eventually go fixed or you go extinct, with some small probability that once you go extinct, you can come back through mutation. And so if we use that approach and look at our data, we see that at the 1-day populations, things are basically the same. The wild type and SPO0A have similar numbers of fixed mutations. There's a lot of variation. But we don't see much evidence of saying that, seed banks matter in this daily line. Whereas for the 10-day experiment, the only populations that have mutations fixed mutations up to day 700 are the SPO0A. So no fixed mutations occur in the wild type on the time scale that we can compare the two treatments. Whereas so that indicates that the rate of evolution increases in the absence of a seed bank. And here the rate of evolution, just the per generation rate of substitutions. And then when you get to 100 day, you really can't do any comparison. There's only one fixed mutation occurring across all replicate populations. So not much can be done with that. So from here, we want to sort of examine the correlation between mutations that can, and how it can be affected by dormancy. So if you just got simple birth-death events, mutations, you get two mutations that can both increase in frequency, or, you know, whereas for if you've got this seed bank in the population, right, where individuals can start in an active state and enter a dormant state, you basically have this demographic structure occurring. And so you would expect that in the presence of this demographic structure, correlation of mutation trajectories between the correlation coefficient of mutations would be smaller here, because mutations can go under different trajectories depending on what demographic state they're in, right? And so we would expect a high correlation between mutations for active and a lower for dormant. And so we see that this is the case where for 1 day transfers, things are, you know, statistically different, but overall the shapes of the curves seem relatively similar, right? If we look at the distribution of squared correlation coefficients between our allele frequency trajectories. Whereas by the time you have this 10-day transfer, the distributions are totally different, where there's a way higher correlation on average for the knockout that can't form a dormant state, which is consistent with our predictions. So for the summarize here, we have evidence that dormancy increases genetic diversity, but this is sort of this nonlinear relationship going on, where the lack of evidence in the 10-day treatment we think is due to the fact that the knockout actually has higher fitness, which relates back potentially to net for mass recycling, what was addressed in the first part of this talk. And dormancy, we find that dormancy reduces fixation events under energy limitation, and that the presence of seed banks reduces the overall correlation structure between mutation trajectories. And so just briefly, we wanted to finally see whether the targets are different for evolution. So we find evidence that negative selection is consistently occurring across treatments, but it is slightly stronger in the 10-day treatment, in a 10-day transfer. So negative selection holds in 1 and 10-day lines, though there's a lot more variance in the knockout. And if we do a comparison, but for the ratio of non-synonymous to synonymous mutations in between 1 and 10-day lines, we do see that significant difference, where for the wild type, it is significantly lower in the 10-day. We don't find that for the SPO0A, and that's likely driven by this just large variation we observe here. But this fact that negative selection tends to be dominant relates back to the first part of this talk, where we found that this DNDS is consistently less than 1 in our energy-limited 1,000-day experiments as well. So we find that, you know, if we examine the actual non-synonymous mutations in the population, we find that they're unequally distributed across genes. So again, that we were able to calculate what's been termed as a multiplicity for each gene, which is just the number of mutations occurring in a given gene that's corrected by the size of that gene relative to the mean size in the population. So it's sort of just correcting for the fact that you have a larger gene, you expect more mutations on average, just simply because it takes up more of the genome. So from there, we're able to calculate genome-wide likelihood of enrichment and do a permutation test and see that, okay, the likelihood of this multiplicity versus the null expectation suggests that it is significant for both strains across all transfer machines. That's easy to reject, though overall it does seem that parallelism is reduced in SPO0A if we actually plot out survival curves of the multiplicity and the fraction mutation. So this longer decay here and the wild type corresponds to a higher bias in terms of where mutations hit across the genome. However, this is likely primarily driven by the fact that there are fewer mutations in the knockout, and we're able to do this by just subsampling our data. So to actually identify the targets of selection, there are a few genes that are significantly enriched in the knockout, right? But if we just try and examine the multiplicity for genes in the wild type against the multiplicity of genes in the SPO0A, only looking at genes that are significantly enriched in both populations, or both treatments here is indicated by the black, we aren't able to reject the fact that the same that the multiplicities for these genes are similar across both treatments, right? We can just, if things were exactly the same, they would fall in this one-to-one line, and we do see deviations, but they're not significant enough. That slope there is not significantly different from one. And that's primarily, I think, driven by the fact that there's just so few genes that are enriched in the SPO0A lines, right? So this big circle here corresponds to the genes enriched in the wild type that can inform a dormant state, and this much smaller circle is in the knockout, right? And that overlap there is the ones that, that actually get hit in both. To sort of just try and visualize this, what we can do is just plot all the genes that acquire more mutations expected by chance in more than one treatment here, right? Here, the first two are 1 day, the second two are 10 day, and the final two are 100 days. And we're able to see that, there aren't really many genes enriched in the 100 day or 10 day, and those that are enriched within a wild type tend to be enriched also for the knockout within that regime. So just removing this master regulatory gene doesn't seem to affect any sort of genome level measure of evolution. So this is through a different approach, but Jay and I have been sort of working on how to best quantify ask these sorts of questions. And so if you're interested, we have a preprint out that's in review that we'll also be likely revising fairly soon as well. So the question for me is how to quantify whether or not these strains are actually diverged at the gene level. And what we eventually realized was that we can just basically use the set of enriched genes and compare them to determine whether or not the genes that are enriched diverged or converged for different treatments. And so you've got some total number of genes in the genome here, right? And you can basically just partition the genes that are enriched for mutations across 4 different groups. The first is just the genes that acquire more mutations than expected by chance in both treatment 1 and treatment 2. The ones that only acquire more than expected by chance in treatment 2, over here for treatment 1, same thing. And then finally, all the rest of the genes that didn't get more mutations expected by chance. And so you just do that 2-by-2 matrix, get an odds ratio, and try to detect whether evolution is divergent or convergent. And we see overwhelming evidence that divergence isn't really happening at the genome level. If anything, strains are consistently converging, even if one can't form a seed bank. So some future directions that I'd like to take this in. My postdoc is going to be primarily computational. And if I ever get back in a wet lab again, I think that something I'd like to do is just get a better estimate on quantities of interest for necromass recycling. So there's a lot of really great work being done, primarily in deep sea sediments. But I think we also need really precise measurements in very, very stripped down sort of cultures to get better estimates on the range of important quantities of interest, such as the number of dead cells required to maintain an existing cell or produce a new cell. And so I think some work I would do if I ever get back in a wet lab is do some manipulable batch and continuous culture type experiments for that. And finally, for evolutionary energy limitation, to actually determine what are the targets of selection under these regimes, which was not our primary goal, but something we tried to look into. I think that a really promising approach is how barcoding has been increasingly being used in evolution experiments. And this actually has already been done for energy limitation for E. coli, where you do those T&C experiments where you have this gene library of mutants, and you just look at the fitness trajectories in different environments and compare them across treatments. And so something like that, but also incorporating the demographic fluctuations that occur under energy limitation, right, where you have this massive bottlenecking event. So I'm a little over time, but of course, got to thank the Lynn Lab. Jay's been an amazing advisor. This is a very old pick. We should take a new one, but I mean, I came into his lab and was talking about experimental evolution, and it was a little done in his lab, but I was completely new to it, and he just was, very supportive and a great advisor that's put up with me like reading all this population literature, which was sort of new, I think, to the lab at the time. But of course, so many people allowed to thank. One to point out is our technician Brett, who helped me get things up and running, and of course funders, Army Research Office, NASA, and Society for the Study of Evolution, and of course help from the people that actually allowed us to sequence these things. And yeah, thank you for listening. And I guess now we get to see some questions.
Great, thank you, Will. Yeah, if you can drop off your screen share and answer them in person. That was fascinating. Really
Thanks.
Interesting work, and I'm really stoked that you've contributed that. So much more evolutionary, I think, than well, at least I was expecting. We do have a few questions in the chat. The first is from Leanne Benning. She says, "Great talk. As far as I understood, the data you showed were for individual species. Did you try combined, and for example, mixing populations with high versus low death rate?"
So sort of a community type question. Yeah, we didn't. I think that would be interesting. I think I would want to try and first figure out like more mechanistically what would go on in that scenario before combining them. So, I mean, if you take the first experiment where we don't have resources, right? We can't apply like a resource consumer model necessarily because there's nothing really initially to grow on. So I think that how the long-term survival of each species would depend on their ability to recycle necromass and whether or not, things are necessarily better at recycling their own necromass or the necromass of a different species. I think that would be really good approach that you could do something like thinking of like how the Sanchez group does a really good job with looking at pairwise combinations of taxa over 2, 3, 4 species. Something like that would allow you to basically, I think, determine any sorts of interesting dynamics that go on. But I haven't done that, no.
Cool. Let's see. Okay, I have a question from Zach Laturner, who he kind of retracted this, but I think it's still relevant because it kind of speaks to some of the strains you were talking about at the beginning before you got to the specific sporulation knockout for bacillus. Have you thought about grouping organisms by long-term strategy of survival, sporulation, et cetera? It seems like these different strategies could affect how you end up modeling them. So I think, instead of, you went to a deep dive on one of those strains that was a sporulator, but what about the others? Are there other sub-classifications, and does that affect how you think about this?
Yeah, I mean, this actually, the bacillus evolution experiment, that is part of this larger experiment that I also ran with Jay that's we're writing up, where we had 5 other non-spore forming taxa that had the same treatment regime going on, 1, 10, and 100 day. And we do see that the bacillus knockout dynamics are very similar to what we see for other taxa that can't form spores. So, I mean, that stuff is coming out, so that there should be some really nice. Hopefully, people enjoy reading that. But we did try to examine it, and a lot of the patterns we observe for the wild type go away when we look at the others. And so I think that those, I think necromass recycling is consistently occurring in all, wild type, the knockout, these other taxa that we examined. But the surviving, they're just not able to survive as well as the bacillus with the spores. You just can't get around that.
Right on. A question from BG Shabulal. Hopefully I didn't butcher that name too badly. Is it possible to identify the genes that are active both in wild and spores during the long-term study?
So like transcriptionally active, you think?
Presumably.
Yeah, I mean, let's see. I think we've got some biomass replicates in the -80 that we flash froze. And so you could, yeah, wake that up and you do some pooled transcriptomic sequencing. This experiment was initially funded off of a large grant that Jay was on with other co-PIs doing different experiments. And that was initially in the proposal, and we just didn't get to it. And so I think that would be interesting, looking at gene expression profiles. But there is a postdoc in Jay's lab right now, Daniel Schwartz, who works on bacillus and also bacillus phage interactions. And he's, I think, addressing some of those questions of differential gene expression between the knockout and the wild type. And so he'll have some interesting work. And I think he's going to try and look at whether or not change in expression corresponds to whether or not those genes acquired more mutations than expected by chance. So it's definitely possible. We can definitely do it. And I think it's going to be done. Yeah. Not by me, though.
Nice. A couple more questions from Nicole Ratib. I might have missed it, but what are the population sizes you're starting with? I'm thinking about how that affects the initial diversity and the evolutionary outcomes.
Right, in the second part of experiment. So it's a 1 to 10 dilution so you basically get about 10 to the 8 to 10 to the 9. I've got the more precise numbers somewhere, but it's a 1 to 10 dilution. And in retrospect, a 1 to 100 dilution is more standard and probably would have been a better choice. But this experiment is initially constructed to be compared towards some larger experiments done on E. coli for Meg Behringer in the Lynch lab. And so they wanted to do 1 to 10, and we eventually were like, okay, 1 to 10 works for us. I think that that might not affect things too much, other than if we get to a much larger dilution ratio, we risk extinction in the knockout, because you basically end up with about 10 to the 3, 10 to the 4 cells total over 10 milliliters of media, so 10 to the 2, 10 to the 3 per mil after 100 days of energy limitation. And so you risk extinction, and then not necessarily in bacillus, but in our other taxa, we do see extinction occurring over this 100-day period. And so we're trying to basically set up a dilution regime where things don't go extinct so much that you can't perform the experiment at all.
Yeah, that's interesting. Tina Rai, thank you for the great talk. You mentioned about the 1,000 days. So I wonder, how do you cope up with the, how do you keep up with the culture for 1,000 days? Could you explain your experimental design again? So, how does one conduct an experiment over 1,000 days? Three plus years, right?
So yeah, for the first part, with that energy limitation experiment, they're in Falcon tubes, and after you've resuspended the cells in the buffer, you just leave them there, and you just take very, very small aliquots and plate them every, I think we had about 4 to 8 days. So you sort of stagger things out so it's not overwhelming. And we did have some contamination in a few lines that we ended up not ultimately using for the data I showed, but overall things were fairly clean. It's much more of having the certainty that you're, I think, in this place for several years, that you're able to justify keeping an experiment of that time scale going. The actual tools and setup is fairly straightforward. You just need to have, I think, dedicated labor to get it done, which is very difficult in itself.
Yeah, and keeping things in pure culture for all that time, I imagine is pretty challenging too. Are these under selection? Did you have them with antibiotic cassettes or something like that?
Well, the first one are in buffer, and it's they're just in buffer. The second one, we had to use a complex media because this was initially going to be compared to some other experiments, and we were doing different taxa, and we had this ultimately defined media that had so many micronutrients, it was impossible not to screw it up when you scale it to large scale media prep. There was a little bit of cyclohexamide we added to the transfers, because even with UV UV light hitting the air intake and the filtering apparatus, and having the flask covered over 100 days with a large enough number of flasks, eventually like one spore would get under there, right under that cap, and then into the culture, and then you have this big like just plume floating on the surface at the end of it. And that became a challenge at first, and we lost I think 5 months of work or something on that. But so we did use a little cyclohexamide, and we ran some fitness assays first to see how much it affects the taxon. It doesn't seem to matter.
Okay.
Yeah.
Well, it's still very, very, very impressive. Anybody who does any amount of culturing is going to be familiar with the struggle of keeping something going for that amount of time. Cool. Well, let's see. I'm gonna let's see. No other. I was going to say vamp for a few seconds to let the lag catch up, because we are on a little bit of a time delay, and sometimes some questions come in right at the end. But it looks like that's about it for now. Thank you very much for your contribution. We'll cut it off here, and everyone that's watching, please stay tuned. We have another micro seminar coming next week. Keep checking our website for schedule updates. And again, thank you very much, Will, and best of luck on your new position. Really looking forward to having you on this side of the country with us.
Oh yeah, and thanks for having me. This is great.
Okay.